Okay, so what I'd like to do today is go over a little bit about the structure and the function of neurons. So what I've done is I've made all of my notes completely available because I think that that will greatly aid in the learning process. I think that you can just listen along and you could screenshot the notes, and that way you don't have to worry about writing or typing what I'm saying. You can really just sit back and listen. So I will do that in all of my preceding videos as well as this one. So neurons are the basic signaling units that transmit information throughout the nervous system. They're going to take in information such as sensory information, such as visual information coming in and hitting the retinas. They're going to make very simple decisions about that information, and then they're going to pass it along to other neurons and other processing areas of the brain. So neurons are not alone in the brain. There are also glial cells. These are non-neuronal cells in the nervous system. They provide structural support, insulation for electrical activity passing down the axon of the neurons, and they also modulate neural activity. This is somewhat of a newer finding, and ongoing research is still looking at this to further elucidate what's going on. We'll go more into depth about glial cells in another video, and we'll briefly touch on them in a couple minutes. So neurons have the same types of things that other eukaryotic cells in the body have, such as the cell membrane, which encases the cell body. The cell body is also known as the soma. And inside the soma, there's other types of machinery, such as the nucleus, the endoplasmic reticulum, cytoskeleton, mitochondria, Golgi apparatus, and other types of things. And all of these structures are going to sit in a salty intracellular fluid. And this is known as the cytoplasm. And within the cytoplasm, there's going to be combinations of ions such as potassium, sodium, chloride, and calcium. Now we'll go more in depth in another video about sodium and potassium because they're very fundamental to the sodium-potassium pump. They're both positive ions, and they play a big role in keeping the intracellular fluid and keeping the neuron at a negative resting potential, usually negative 70 millivolts. Chloride is used in iPSPs, inhibitory signaling, and the calcium is a positive ion that's usually used in the axon terminal to release the neurotransmitters from the vesicle and pass along through the synapse to bind to receptor sites on the postsynaptic cell. So another important point is that neurons sit in a bath of extracellular fluid, which is made up of ions as well, both positively and negatively charged. There's going to be proteins that are positively and negatively charged sitting in the extracellular fluid as well as the intracellular fluid, which allow for the positive and negative charges. So like I said, the resting potential of a neuron is around about negative 70 millivolts, and the extracellular fluid is significantly more positively charged than is the intracellular fluid. Now, because this is the case, it supplies the neurons with a constant source of energy, shall they ever need it, and they do, to actually fire. So there are two main parts to a neuron that everyone thinks of. They are the dendrites and the axons. And I actually believe I have a picture here. Yes. Okay, so this is a classic depiction of a neuron here. This is going to be your soma, your cell body, your nucleus right there. And these are going to be your dendrites. So these are the dendritic tips. You can see right here there are actually dendritic spines that would protrude right here that you can't really see. And on these dendrites are going to be receptor sites that are going to pick up neurotransmitters released from the presynaptic cell. The presynaptic cell, you can imagine, is right here, and there's going to be a synapse here. Neurotransmitters will travel through the synapse. They will bind on the receptor sites, and this will be input to this neuron. So what happens when a neurotransmitter binds here is that it allows positive ions to come into the cell. Now, all of this positive influx here will ultimately culminate at the axon hillock, which you can just think of as a gate, and if the threshold is broken, if there is enough depolarization, then the gate will open and that activity will go down the axon. Now, going down the axon is known as an action potential, and we will go more in depth into that later. And so the action potential will fire down the axon. It will go to the end bulb where the neurotransmitters will be released and they will bind on to receptor sites of a postsynaptic cell. So this is the dendrite. These are the dendrites here. This is the soma, and this is the axon, and your axon terminals here. So what you can see here is that the axon is made up of round, white, fatty type substances here in casings, and that is going to be your glial cells. So here you can see this would be an example of a glial cell, and it's wrapping itself around, insulating the axon. This facilitates the electrical transmission of the action potential. Now I believe these would be so it actually says it up here. These would be oligodendrocytes. They're going to be made up, or they're going to be insulating neurons in the central nervous system, and Schwann cells will be doing the same type of thing to neurons in the peripheral nervous system. Other than you can see here that this glial cell can also insulate other neurons, whereas in the peripheral nervous system this isn't the case. So we will talk more about that in the future. So that, I believe, will conclude our talk here about neurons and the structure of neurons. In the next video, we will go into neuronal signaling.